Science in the morning, science in the evening, science at supper time. When science is on the internet, you can have science anytime with Mr. Francis.
Hey everybody, this is Mr. Francis here, just recording a little video for you since I can't talk to you in person. Uh, some of you have started this already, some of you have not, but we're going to move forward with it and make sure that we do what we got to do. Okay, here we go. So today we're going to talk about DNA structure, function, and replication, and it's pretty interesting stuff. Let's go. So first off, what does DNA stand for? The term DNA is really short for something called deoxyribonucleic acid. Deoxyribonucleic acid is the stuff that makes you you. It's your instructions for life. And we name it deoxyribonucleic acid because one, it's a nucleic acid, and two, it's got a sugar called deoxyribose. We name all nucleic acids based on the sugar that is found inside of their monomers. What is the function of DNA? Well, it stores genetic information. It's a code of life. It's a whole bunch of letters that make you you. And if we change any of those codes, we change you. You get your DNA from your mom and from your dad mixed up, so half and half. And if you take a look at this picture that's on the screen here, you can see that the father has two shades of blue, and the mother has a pink and a red. Now, their offspring, which are down here, you're going to see that it's a mix of both blues and both pinks for each child. This is what child 1 and child 2. This is because of something called the law of independent assortment. So when a sperm or an egg is created, we don't know what DNA is actually going to end up in each one. And it mixes up quite a bit. That's why everybody kind of looks like their parents, but sometimes they look a little bit different. What is DNA made of? Well, first off, as we learned earlier in the year, DNA is a nucleic acid, and a nucleic acid is a type of organic compound. We learned that organic compounds all have different types of monomers or single pieces located inside of them. And the monomer for DNA is called a nucleotide. A nucleotide, if you remember, is made up of 3 different parts. We have a phosphate group, a sugar, and a nitrogen base. The sugar in DNA, again, is called deoxyribose, and that's why it's called DNA and not something like RNA. So when we talk about the structure of DNA, it's a whole bunch of nucleotides that connect to each other. And they form almost like a ladder, if you will, that goes up. On the sides, we've got the sides of the ladder, and in the middle, we've got these rungs. That'd be the part that you walk up and down. The sides of the ladder are made of a sugar and a phosphate. It's called the sugar-phosphate backbone, and they all connect to each other as they go up. Inside the middle of the DNA, the rungs, the part that you would walk up, is what we call the nitrogen bases, or nitrogenous bases. Let me show you a picture here. So you can see here we have a phosphate and a sugar, phosphate, sugar, phosphate, sugar. And these highlighted in purple here, this is the side of the ladder, or the sugar-phosphate backbone. In the middle, you can see the nitrogenous bases hanging out. If we were to put them next to each other, you would see, again, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and the same on this side. In the middle, AT, TA, Cg, and Gc. These are the bases, or the nitrogenous bases. One thing you might notice, though, is that these sugars, they kind of point in one direction or the other. And what we're talking about is something called anti-parallel. Anti-parallel is just telling you that one's going up and the other's going down. I can't quite get my arm in there for that, but they are going up and down in different directions. This helps us when we replicate the DNA to make sure that it replicates in the correct order. The shape of DNA is what we call a double helix. Okay, here you can see that double helix also in the background of this screen that you're looking at. Okay, this double helix is like a ladder that's been twisted on both ends. So imagine that I held out a ladder to you and you were holding the other one, and we both started twisting it. Okay, and as we twisted it, that would give us that kind of spiral staircase shape. That is what we call a double helix. In the middle of our DNA, the rungs of the ladder, the parts you would walk up and down, these are the nitrogenous bases. You have 4 of these, 4 nitrogenous bases that DNA includes. We have guanine, adenine, cytosine, and thymine. Or, usually we refer to them as G, A, C, and T. Now, when we talk about these, we have to talk about complementary base pairing rules. Okay, A always goes with T and G always goes with C. So adenine and thymine are always connected together, and guanine and cytosine are always connected together. They don't really look like this, the green and the pink and the blue and the orange, but it really makes them, I don't know, makes it help you understand. They fit like a lock and key. Adenine can only fit with thymine, and guanine can only fit with cytosine. They can't be mixed up in any other direction. One way I like to remember that is with apple tree, garbage can. Apple tree, garbage can. Say it with me. Apple tree, garbage can. I mean, I guess you could get weird and you'd be like, tree, apple can, garbage, but that just doesn't make any sense in my mind anyway. So I always like to remember apple tree, garbage can, and that will help you out a lot in your studies. When we talk about the bases, you might also have noticed that some of them only have a single ring. These are called pyrimidines. Here's a pyramid, I like to remember that. And we have some that have double rings, and these are called purines. What I like to remember with cytosine and thymine, they both have the letter Y in them, and so does a pyrimidine. And if there's a single ring and there's a single point of a pyramid, if I was to do a ring toss, let's say, it would land right on top if I'm a good shot anyway, and I would win. Purines, on the other hand, are two rings, adenine and guanine. And I like to think of pure love, like a set of wedding rings here, because it's two rings involved in that. Now, when we have the sides, the sugar phosphate base, backbone rather, and we've got the bases in the middle, what we have holding those bases together are what we call hydrogen bonds. Okay, hydrogen bonds are what hold the bases together. Some have 3 hydrogen bonds, as you can see between G and C, and A and T only have 2 hydrogen bonds. These are pretty weak bonds that can be broken down by some enzymes to help us replicate DNA. So, speaking of DNA replication, how do we make more DNA? Well, we replicate it. Why do we make it? Because we constantly need more cells. Right now, your cells are dying and falling off your body, being digested. We constantly need to replace these cells. Okay? And we need more DNA to do that. Because if we don't have DNA, you can't get more cells. It went backwards there, but that's okay. So the word replication, it's when we make an exact copy. Not something different, but an exact copy. So for instance, if I took one dye and I replicated it, I would have 2 exact copies of that dye. Or if something that's not replicated but close, if I have this thing, it's called a life counter, it's for a game, and I made this, you can see that they're different, okay? They're not exactly the same. So that's not technically replication, because replication needs to make an exact copy. So again, DNA is copied through a process called DNA replication, and we use some things called enzymes. If you remember, enzymes are types of proteins that speed up chemical reactions. Without them, we would die. It's really important that our enzymes are working correctly so we can function correctly. So the 2 different types of enzymes that we use in DNA replication, first one is DNA helicase, and this one is like a zipper. And what it does is it spins in a circle and unzips DNA from all the way to the bottom, all the way up to the top. And I like to think of a helicopter spinning in circles and rising up. DNA polymerase, the other one. If you remember, poly means many. What it's doing is it is adding many base pairs right behind the DNA helicase at something called the replication fork, or like a fork in the road. And what it's doing is making sure that it's adding the correct base pairs in the correct order, and it's gotta match, because if it doesn't, that's not good. So it checks for errors as it is going through and adding those base pairs behind the DNA helicase. Here's a quick image of that. This is from our textbook from last year. Here you can see the helicase unzipping the DNA as it moves up. Here's our replication fork, almost like a Y. And as it moves up, the DNA polymerase follows up behind it and adds base pairs. Now this gets really, really interesting, because one side is really, really easy, and the other side, it's not really easy. We're not going to get into the lagging strand versus the good guy strand, I guess you can call it, the easy one. But just know that as it unzips, we're adding base pairs to each side. And then what we end up with is 2 strands of DNA that are exactly the same. Each one, as you can see in this picture, is made up of new DNA and old DNA. And we call this semi-conservative because we're not wasting anything as we make new DNA. Here's another image of that. Okay, so here's the helicase unzipping the DNA, and as it unzips it, we have the DNA polymerase, adding base pairs. Okay, this is the real easy one. It follows right behind the helicase and it goes in the same direction. While on this side, what we call the lagging strand, or the not so fun strand, it has to move backwards in the other direction. And to do this, it has to have all kinds of promoters that click in there and Okazaki fragments. And it gets really, really intense. We're not going to watch this video because I don't think I can do that, and that's okay. And we're not going to watch this one either, but you can click on this because this PowerPoint is in your Google Classroom. So feel free to move forward and watch these two videos. This one's really good. Just don't have your speakers really loud, okay? Well, guys, that's it for me. Hopefully you found this helpful. It's my first time trying to make a video, so bear with me. I hope it was okay, and let me know in the comments below this if you liked it or not. Okay, have a wonderful day. Well, night, whatever, whenever you watch this, whatever. Just be safe. Wash your hands. Have a great day, guys.